Point mutations typically deal with individual bases or groups of bases. A much larger scale mutation would be a chromosomal mutation, and some of the same terms are used. You can see over here, if a gene is removed, that's called a deletion. And in this case, you're removing an entire gene. So unless that happened to be a bad gene, that's usually not very good. A different type we have is called a duplication. That would be where you end up getting two of a gene. And if it happens to be a protein that's really important that your body needs, that could be good, or it could be bad if there's too much of it that goes on there as well. Sometimes we have inversions where genes get flip-flopped, and that could affect the entire sequence of the gene, or if it's pretty intact, it could still code a similar way. It also could be a bigger problem, though, down the road if the genes don't line up and crossing over occurs like we saw in meiosis, and the crossing over occurs kind of in the middle of a gene. You can have some real issues there. Insertion is when a gene from one chromosome gets inserted into another, and obviously this can cause some problems because now the chromosomes won't be balanced, and you could have an organism that has an allele missing or an extra allele, much like in the duplication stage. And then the last type we have over here is called a translocation, trans meaning across, so it's changing locations, and where you can see there's some flip-flopping here, so it's somewhat similar to the insertion except for both sides are inserting, and once again you'll have slightly different chromosomes, and so they won't be able to line up perfectly in homologous pairs. So the insertion and the translocation will affect your homologous pairs, where the deletion, duplication, and inversion will mostly just affect the gene production. So those are 5 types of chromosomal mutations, and that's about it.